Hello friends, good morning all of you. Welcome to my channel, and those who have not subscribed, please subscribe to my channel for new videos. So today's topic is the limbic system. So the sense of smell or olfaction, one of the primitive form of sensory modality, is highly developed in macrosmatic vertebrates, and it monopolizes major portion of the forebrain in early phylogeny. Hence the forebrain was considered as the smell brain. So the name is the Rhinen-Keflon. Rhinen-Keflon. So it also, the olfactory system not only receives just a smell but also activates the other neuronal systems for emotional behavior in the form of increased salivation, gastrointestinal motility, movement of procuring food, and aggression for self-defense. So therefore, sex urge for mating, railing the younger generation, and therefore a basic drive for preservation of the individual and the species. So let us discuss about the anatomical components of this limbic system. So coming to the anatomical components of this limbic system, moving to the anatomical components. So first one is olfactory nerves. Olfaction, the smell, the nerves which carries the smell impulses from the nose, and they takes to the olfactory bulbs which are located in the anterior cranial fossa. Then from the olfactory bulbs they go posteriorly through olfactory nucleus, olfactory tracts, and the retics and their terminations. Then we are going to discuss about the pyriform lobe, that includes the uncus and entorhinal area. They acts as the primary olfactory area and association olfactory areas. Then third one is amygdaloid body and its efferent pathways. They are striae terminalis and ventral amygdalofugal fibers. Then the hippocampal formation comprising indusium griseum and longitudinal striae, gyrus fasciolaris, dentate gyrus, and hippocampus. Efferent projection fibers of the hippocampus forms the alveus, fimbriae, and fornix. Not only these four, but other structures are limbic lobe, which includes the septal area, cingulate gyrus, parahippocampal gyrus, and the latter lies in continuity with the primary olfactory cortex of pyriform lobe. The last one we are going to discuss is the hypothalamus, anterior nucleus of the thalamus, habenular nucleus, interpeduncular nucleus, midbrain tegmental nuclei, striae medullaris, thalami, fasciculus, retroflexus, and medial forebrain bundle. So these are the structures we are going to discuss. So the first one which we are going to discuss is the olfactory pathway. So olfactory pathway, it includes olfactory nerves, olfactory bulbs, anterior olfactory nucleus, olfactory tracts, and their attachments. So these are the structures we are going to discuss one by one. So let us see that. Yes, coming to these olfactory nerves, these are derived from the central process of the bipolar neurons. So you can identify the neurons over here. So these are the bipolar neurons. So this bipolar neurons, it is having central process and the peripheral process. So these are derived from the central process of bipolar olfactory neurons, which are situated in the olfactory zone of the nasal mucous membrane, involving the roof and the adjoining septal and lateral walls of the nasal cavity, and covering an area about 2.5 centimeters square. The peripheral process of the olfactory cells appear at the surface as sensitive olfactory hairs. Sensitive olfactory hairs, which responds to volatile, water-soluble, and lipid-soluble odorless chemical substances. So the olfactory cells are supported by tall columnar sustentacular cells. Can you identify this? So these are nothing but the tall columnar sustentacular cells which are provided with microvilli on their free surface. So as earlier we have seen that almost there are 10 million bipolar olfactory cells are present in a man. Then let us discuss about this olfactory bulbs. So what are the structures present in the olfactory bulbs? So in the nasal mucous membrane, we have seen the tall columnar sustentacular cells. Here you can identify these are all nothing but the tall columnar sustentacular cells. Then this roof, as we have seen that the cribriform plate of ethmoid bone, which is it is having the perforations through that these olfactory nerves are passing. Then moving to the above structure, above the cribriform plate of ethmoid bone, we can identify the olfactory bulb. So you can identify this is nothing but the olfactory bulb. So each bulb is an ovoid mass of gray matter and situated on the orbital surface of the frontal lobe at the rostral end of olfactory sulcus. On the rostral end of olfactory sulcus. So the olfactory nerves reach the undersurface of the bulb, which is continuous behind with the olfactory tract. So this is nothing but the circular one, this is nothing but olfactory tract. Anterior one, this is called as olfactory bulb. So this bulb is consisting of the mass of gray matter. So what are the structures are present here? From the periphery to the central core, we can identify a layer of nerve fibers derived from the ramification of olfactory nerves. Then inside we can identify a layer of synaptic cleft. Yes, here you can see that a layer of synaptic glomeruli found by the arborization of the central process of bipolar neurons and the dendrites of the mitral and the tufted cells. So these blue color cells are nothing but the tufted cells. And other structures here you can see the mitral cells, the blue color one here, the tufted one. This is the mitral cells. These two are the mitral cells. So these mitral cells and the tufted cells, they are forming synapse with the central process of the olfactory neurons. Here you can identify that. So this synapse which we can identify is where we can identify this synapse is at the periphery part of olfactory bulb. We can identify the synapse. Then the external granular layer consisting of external granule cells. Let me show you this external granule cells. Here you can identify this green color one that is nothing but external granule cells. Okay, so and even the tufted cells, they are also extending into the central process of that. So the both cells are present, the granule cells and as well as the tufted cells, mitral cells, they are also extending into that. Then the dendrites of the tufted cells enter into the synaptic glomeruli and their axons pass through the olfactory tract. So here you can see that. I'll show you. The dendrites of the tufted cells, they are extending. This blue color one, they are nothing but the dendrites of the tufted cells and the mitral cells. They are extending posteriorly in the olfactory tract. So the few axon collaterals make synapses with the external granule cells. Then a layer of mitral cells, this blue color one, this is nothing but the mitral cells. The mitral means it is appearing like the bishop's cap, the mitral cells. So these apical dendrites of the mitral cells form synaptic glomeruli and the basal dendrites make synapse with the internal granule cells. So these mitral cells, they are forming synapse with the granule cells, internal granule cells. Then the axons of the mitral cells arise from the center of the basal zone and form a chief constituent of olfactory cells. So in the olfactory tract, what are the structures we can identify are the dendrites of the tufted cells and the dendrites of the mitral cells, which we can identify in the olfactory tract. Then a layer of granule cells also we may see in that. Then moving to the next structure is olfactory tracts and the pyriform lobes. So olfactory tracts and the pyriform lobe, each tract extends backwards from the posterior part of the olfactory bulb and lodges in the olfactory sulcus, so which is at the orbital surface of the frontal lobe. Then that sulcus, if you just take a section, it appears like a triangle in shape. So it is formed mostly by the axons of mitral and tufted cells of olfactory bulb. A few centrifugal fibers arising from the opposite olfactory bulb and anterior olfactory nucleus that enter the tract through the anterior commissure. So here you can see that. So they are entering posteriorly and reaches to the anterior commissure. We already have discussed about the anterior commissure in the white matter of cerebrum, where the right and left cerebral hemispheres, which are the fibers which are joining with each the right and left cerebral hemisphere through this anterior commissure. Okay, so this olfactory mitral cells and the tufted cells, the dendrites, which are extending posteriorly and reaches to the anterior commissure. So traced behind, the olfactory tract approaches the anterior perforated substance. These perforations are nothing but anterior perforated substance, where the fibers of the tract flatten out to form olfactory trigone. This entire area is called as olfactory trigone, where we can identify this olfactory tract, which is going to divide into medial and lateral olfactory striae. Medial and lateral olfactory striae. Okay, so that is about the olfactory trigone, medial and lateral olfactory striae, and anterior perforated substance. Let us discuss about the medial striae. Medial striae. Coming to this medial striae, this is medial striae, and here you can identify the lateral striae, and sometimes you may identify intermediate striae also. So let us discuss about the medial striae. Medial striae passes along the cranial border of anterior perforated substance. Anterior perforated substance. So in front of the cranial end of the anterior perforated substance and appears in the paraterminal gyrus. Paraterminal gyrus. And in front of it is exactly present in front of lamina terminalis. Here you can see that lamina terminalis just below the corpus callosum. Here you can see the genu of corpus callosum, and here you can see the rostrum of corpus callosum, and rostrum of corpus callosum, it is extending as the lamina terminalis. So just in front of this, here you can see the paraterminal gyrus. So this here the medial striae is accompanied by a thin sheet of gray matter that is called as medial olfactory gyrus, and by the diagonal band of Broca. So most of the fibers of the medial striae end in the paraterminal gyrus. So a few fibers, however, pass through the anterior commissure. Here you can see that anterior commissure. So most of some fibers, they pass through the anterior commissure and connect with the neurons of opposite anterior olfactory nucleus and olfactory bulb. Okay, so they are passing through the anterior commissure and joins with the opposite side olfactory bulb, olfactory nucleus, and the olfactory bulb. Then the fibers of intermediate striae. Here you can see the intermediate striae. So this is the intermediate striae. So the fibers of intermediate striae, if present, it terminates in the anterior perforated substance. Then let us go and discuss about the lateral olfactory striae. So this is the lateral olfactory striae. So the lateral olfactory striae passes along the anterolateral border of anterior perforated substance, and then turns abruptly backward and medially to be continuous with the gyrus semilunaris. Let me show you this gyrus semilunaris. Okay, here you can see that this is nothing but the gyrus semilunaris, which is incorporated in the corticomedial division of amygdaloid body. Here you can see that amygdaloid body. Yeah, here you can see that. So this is nothing but amygdaloid body. Then the lateral striae is accompanied by a thin sheet of gray matter called as lateral olfactory gyrus, which is continuous behind with the gray matter of gyrus ambiens. So this gyrus ambiens and the lateral olfactory gyrus, lateral olfactory gyrus form together the pre-pyriform region. The latter is continuous with the entorhinal area, that is area number 28, in the anterior part of parahippocampal gyrus. So here you can see that parahippocampal gyrus. In front of this parahippocampal gyrus, here you can identify that prepiriform lobe, prepiriform region. So the prepiriform region, periamygdaloid region, and entorhinal area are collectively known as the piriform lobe. So what do you mean by piriform lobe is? What do you mean by piriform lobe is prepiriform region, periamygdaloid region, and the entorhinal region, entorhinal area, are collectively known as piriform lobe. So which is represented externally by the uncus. So here you can see this is the uncus. So it is externally we can identify the uncus and anterior part of parahippocampal gyrus. So anterior part of parahippocampal gyrus. So entire this region is called as piriform lobe. So the fibers of lateral olfactory striae, fibers of lateral olfactory striae make synaptic contacts with the neurons of anterior perforated substance, lateral olfactory gyrus, and gyrus ambiens, and corticomedial division of amygdaloid body, in the gyrus semilunaris. Okay, so these structures, you remember, this is nothing but the primary olfactory cortex. Then from the primary olfactory cortex, the fibers are projected to the secondary olfactory cortex, that is called as association cortex. So which includes the entorhinal area, that is area number 28. Then primary olfactory cortex also projects the fibers to the basolateral part of amygdaloid body. Then dorsomedial nucleus of thalamus, hypothalamic nucleus, and to the septal areas. So other things we are going to discuss one by one. In the next part, we are going to discuss about olfactory reflexes and as well as amygdaloid body. So till this, we'll have just a short revision once again. Here you can see the olfactory neurons, bipolar neurons. Then above it we can identify two types of neurons we can identify. One is mitral cells and the tufted cells over here. Then the dendrites are extending back. So these cells are present in the olfactory bulb. The dendrites are extending into the olfactory tract. Then after that, it is going to divide into medial and the lateral olfactory striae. So the medial one, which is going towards the anterior commissure, that is in front of the paraterminal gyrus, and then lateral one is which is going to the lateral olfactory gyrus. Then it is reaching to the piriform lobe, which is consisting of prepiriform region. Then we can identify the parahippocampal gyrus, and then amygdaloid body. Okay, so this is a brief revision of this class. Okay, in the next class we are going to discuss about mainly about amygdala. Thank you, students. See you soon.